Hello.
Did it work better this time?
I can see everything clearly and hear everything well.
No, that's a good thing, right?
For my dumb ass, it is. Thank you. Self-deprecating isn't bad.
All right, we'll wait a few more minutes to see if we can get some other people, and people like to join a little bit later, so that light is really bright back there. I don't even really need it on. I'm gonna maybe go turn it off.
Since I'm late for stuff on occasion, what time is the test tomorrow?
The usual lab time.
So 11 or 11:15 or whatever.
Yeah, 11:30. I mean, if everybody is in, or relatively everybody is in lecture, which I don't know if you guys are planning on going. I'm not going to judge either way because we're going to go over the last chapter, but um then we can just go up and get started. I mean, you guys know what to expect now. It's the same as the other one. So, we're going to rotate and scantron and all that stuff. So if people did come in late, just pick up in a blank spot and go from there. So it's not a big deal, but
Thank you.
yeah. Ripley, what are you doing? There's a ladder over by the water bowl. She's scared of everything. So ladder just being there is terrifying. Come on. I know. You're on TV. Yeah. You're on TV. Okay, let's see. Check how many people we've got. Mhm. Go ahead and get started with this. So I'm going to start this. It's an actual live Kahoot. So when it starts, you guys can join it either like separate window on your computer, on your browser if you want to, or on your phones separately if you want to just kind of do both at the same time. But I'm just warning you guys that it's going to be an actual, like we would do in class, play along sort of Kahoot. So let's go ahead and get started. Oh, and this one, it's like the other one, it's going to have a few slides with like where I'll go over the information too. So just a couple minutes on this and then we will actually get started. So just want to give people a chance to join. I don't know why I put this so far away. All right, I'm going to go ahead and get started with this. It might be a little off from my screen. That's okay. Just go with it. All right, yeah, so pretty much everything we have learned so far in lab is going to be considered fair game. I'm also, to be fair, going to let you know that I'm not going to ask you any questions about the eukaryotes. I'm not going to have any in any of the pictures, and I'm not going to be actually asking questions in the multiple choice questions, like about nucleus and stuff like that. Now, that stuff will probably be on the lecture final, so keep that in mind, but it's not going to appear on the lab final. I know that was some of the stuff that was like, I don't know, I don't want to say the hardest, but the most obscure or whatever. So yeah, that's not going to be on there. Hint, if you guys remember like what the slides were like in the last lab, the lab midterm, and of course, I don't have any like pictures of that to show you, but it's the same slides. I didn't make different ones. So if you recognize those slides, that should be helpful too. Okay, so we'll have the 26 stations, we'll have the microscopes and the tubes. There are only, I think, 4 microscope questions, like actually like involving the microscope. Or 4 stations involving it. Most of it is not that. Most of it's going to be the tubes. So just don't focus too much on the slides. Try to remember that you have learned some of that stuff. So it should just be pretty easy recall. And then focus a little more energy on the tubes. That's what I would do. Color changes and everything like that. So it's 50 questions, multiple choice. We'll be doing it on Scantron. So you will need to bring your number 2 pencil, and you will be allowed the 1 minute per station thing. But remember, you can, at the end, go back to whatever station you need to. It's going to be the same setup as the last time, so you should have plenty of time at the end to go back and answer questions if needed. All right, we're going to start with culturing basics. And again, just going to be inoculation, growth, that sort of stuff. So what is most commonly used to inoculate cultures? Yeah, that's a gimme, I feel like. I will tell you guys, like, when we get into the tubes, because the tubes are like at the end of this, because that's what we learned last, but some of the questions could be, how was this media inoculated? So you would have to know which ones were stabbed or which ones were streaked on the slant or both or whatever it is. So in which quadrant of a streak plate would you expect to find the most single isolated colonies? Yeah, that's going to be in quadrant 4, where they're the most diluted out. Okay, so as far as culture basics goes, again, this is mostly review. Aseptic technique, kind of just be aware of how it works and what you need to do to achieve it. Lab safety basics, like broken glass. Types of media, so it's going to be like broth versus slant versus the butt versus the auger. Plates, yeah, or like even the semi-solid like we use for motility. And then different ways to inoculate that media. So based on whatever kind of media you would be given. Now, if you were to see a slant, we know, if you can remember back to the TSIA, that's that orange slant, that we did a stab and a streak on that one. I feel like that's the only one that had 2 of it going on. So be aware that that's like the special situation. Okay, into the stains. What is the first step in the gram stain procedure? Yep, that's going to be crystal violet. So I'll probably go over at the end, but just because we're already talking about the gram stain, crystal violet, it sticks in the peptidoglycan cell wall. So it really gets caught up in the thick cell wall peptidoglycan of gram positives, and not so much in the gram negatives, which just have a single layer of it. And then we will fix it into place with a mordant. The mordant that we use in this particular case for the stain, gram stain, is iodine. It basically fixes whatever crystal violet is stuck in there into place. So that when we rinse with alcohol, that'll remove the outer membrane of the gram negative and remove any unfixed crystal violet. So when you do that rinse with the alcohol, if you were to look at the gram positives, they should still be purple because the crystal violet is stuck in place. Whereas after the alcohol only, the gram negative should be translucent or clear, right? Until we counter stain. And the counter stain is safranin, which is a reddish pink color. Okay, what color do properly stained gram negative bacteria stain in the gram stain procedure? I feel like that should be pretty obvious since we just talked about that just a second ago. Right, so that is going to be red from that safranin once we get that counter stain on there. So, Sassy, what's the name of this particular staining procedure? Right, so that's going to be the endospore stain. You can see like those little endospores were the little bubbles that were kind of red on the outside, but they were that greenish on the inside. The vegetative cells will turn red from the safranin, which is again our counter stain. Your primary or first used stain in the endospore stain is going to be malachite green. Okay. Let's see. Which of the following bacteria will be acid-fast positive? So this one, I chose names or whatever that kind of could trip you up if you weren't very familiar with the names at the time. So Mycobacterium, that's the one associated with the acid fast. That's going to just be, memorization for that one and not getting tripped up seeing similar words like mycoplasma or micrococcus, that sort of stuff. So Mycobacterium. And to be clear, myco in that case comes from the mycolic acid that gives it that waxiness on the outside. That's very helpful and it's like protective. What is the name of the primary stain used in acid-fast staining? Okay. All right, 7, 2, 5. Okay, sorry. I had a question. So what is the name of primary stain used? That's going to be carbofuscin. Yeah, so that's just going to be remembering what's going on here. So in the acid-fast stain, we start with carbofuscin. It actually is able to interact with that waxy coating on the mycobacterium, including the mycolic acid. And since other bacteria don't have that, it gets into that waxiness and then causes the pink to stay there. And then after we do our rinses with the acid alcohol, we'll go back and counter stain with methylene blue in that case. So the negative cells will be blue. All right, after we heat fix the slide, when do we use heat again in that acid fast procedure? All right, whenever we performed our acid fast procedure, that's going to be 5 minutes with carbofuscin. And then it's going to be after that, just straight moving into the rinses. So the acid, alcohol, and all that, and then counter staining with the methylene blue. So there's no heat once we've done that heat fix in this one. So it's kind of a trick question, but there is a method that exists that involves heat for acid fast staining. We did not use that procedure and we did not learn it in the lab. So what color is a negative acid fast stain? Right. Yes, so that's going to be blue from the methylene blue. And the reason I ask questions about the acid fast is because when I worked in Southwest Integris in the lab, we routinely got samples to test for acid fast bacteria from sputum samples usually. So that is something that if you're working in a hospital, especially in areas where there are maybe homeless people in situations like that, that you are likely to see actual possible cases of tuberculosis and then wanting to use the acid fast stain in order to test for it. So that one's kind of a useful one to know how it works. All right, in which staining procedure do we use steam? So in this case, this is one where we do use heat after actually having done the beginning part of the procedure. All right, that's exactly true. That's the endospore stain. That's because we're going to be trying to force the malachite green into the endospores themselves, and it takes 10 minutes of steam in order to do that. I don't know what's going on. All right, so we're going to want to know the procedures. Here I'm wanting you to know the order of the stains that are used, and also how those stains work if we learned about that. Like I just talked about with the malachite green, as far as that using the steam to get it in there, but that it's important because it will actually be able to get into the endospores and not into vegetative cells, that sort of stuff. We've already gone over and over with this on the gram stain, so I don't think we need to touch on that. But yeah, I would hint, I would know the capsule stain, what it looks like, what a positive looks like, how we do the staining procedure with that. So with the capsule stain, remember that's going to be starting off with the Congo red. We don't heat fix that one. So that's when we put the little drop of the Congo red and we mix the sample in with it, and then we spread it out like a blood smear using another slide, and then let that air dry. And then we add the Meneville's drops all over it, and let that sit for a minute, and then we dunk it in the Copland jars. And after we've done that, then you can blot it and then look at it under, but we never heat on that one. So that's important to remember, because if we were to heat that, and because we have to start with mixing it with the stain to create the background, because it's a negative stain that stains the background. So that would cause the cells to shrink up, and that could create a false halo around the cells. And that's what we're looking for as far as identifying whether the organism has a capsule is if it has a halo surrounding the cells. So yeah. Yeah, would know it. Would know capsule. Or Congo red was the primary stain there. And then we'll add Meneville's later on, but it's not the primary technically. And it's a negative stain. Okay, differential and selective media. Details. That just means I want you to know the details. What makes it differential? What makes it selective? Is this media differential or selective or both or neither? So this is going to be dealing with, there is a packet of like the summary of all of the media that we did use in the lab that lists what makes it differential or selective or neither. And that was 6 different plates. Remember, that's going to be like EMB, McConkey, and that sort of stuff. I'm sure you guys remember those. All right, what type of media is this in the picture? Hi, Ripley. Yep, so this is going to be EMB. So eosin methylene blue, and that is in fact for that media. Eosin and methylene blue are going to be the selective agents in that. It is also differential for lactose usage. We will turn pink if we do use lactose, and we will turn that metallic green if we like aggressively use the lactose, like we have seen in the past with E. coli. Every lab should have gotten to see that. But I'm not testing you on what chemical causes that lactose to turn pink. So keep in mind, the lactose is not the differential agent. It is differential as far as it can tell us whether it uses lactose. I didn't tell you the differential agent on how we determine the lactose because it's some ridiculous situation that I don't need you to know, FYI, but it is both. Okay. What is the selective agent in this media? That's the EMB that we just talked about. EMB, man. So yeah, so once again, eosin methylene blue is going to be the selective agent. So the thing that's going to allow gram negatives to grow and inhibit the growth of the gram positives. All right, which one are we looking at here? As best as you can tell from the picture. All right. So in that picture, we have McConkey agar. The thing that is giving away the answer to this one is the fact that the growth on the surface is pink, like the actual growth looks pink in that picture. And so in McConkey, if we have lactose usage, again, just like we saw with the EMB in the previous one, if we just have regular routine lactose usage, it'll turn, the growth itself will turn pink, right? The media has that sort of reddish translucent color, which, to be fair, so does the mannitol salt agar, but we wouldn't have seen the pink growth. That's not normal for regular bacterial growth. All right, this could be more than one answer. So is this media selective or is it differential? Talking about McConkey here. Okay, so McConkey agar is both selective and differential. It's selective. It has It's probably going to ask you this in the next question, but I'm going to go ahead and say it just for, clarity's sake. It has bile salts in it, and that helps for enteric bacteria, which are typically gram-negative, to grow on the media, and it helps select away the gram-positive. So the gram-positive cannot grow on the bile salts. Typically favors enteric bacteria, which are gut bacteria. And then we have the differential side, which is looking for that lactose usage, which we talked about in the EMB a little bit. All right, what media is this? All right, so that is mannitol salts. Agar starts out that red. The pH indicator in this, like we see with a lot of our tubes that we use, right, is going to be phenol red. It turns yellow in the presence of acid change, and it'll turn pink in the presence of the alkaline change. So in that case, we saw yellow. That was turning it. That means we're producing acid from breaking down the mannitol in the media. That typically is going to be attributed to Staphylococcus aureus. So not always, but yeah. It has high salts in it, like actual salt. That inhibits the growth of the gram-negatives typically. It favors gram-positives, but particularly Staphylococcus. They're hardy. So what is the pH indicator that was used in that medium? I just said this one. Of course, this one's probably so familiar by now to you guys. All right, so this one is going to be phenol red. Remember, yellow for acid and pink for alkaline. Okay, so the things that you would want to know for the differential and selective media. Know which ones are selective and which ones are differential, or both, or neither, right? If we're talking about things like the blood auger, remember that the blood auger, it is differential. It will allow us to determine hemolysis, right? Beta, alpha, or gamma hemolysis, but it is not selective. It is actually going to have ingredients that help certain bacteria grow because it gives them, certain nutrients that they can't make for themselves, which we call growth factors. That makes a blood auger a enriched media and helps fastidious organisms grow, and they're the ones that need the growth factor. So there's your words. I said them again. Let's see. Know what the selective agents are and be able to tell me why they are selective. So like we were talking about with the bile salts being something that inhibits gram-positive growth versus allowing enteric growth, because those are the ones that are adapted to live in that environment. Be aware of color changes and what those would mean on each media. Be aware of pH indicators. Blood auger, Staphylococcus media, we didn't talk about those in these pictures. I just mentioned blood auger, thankfully, but Staphylococcus media, it grows staph. I mean, I'm not going to ask you about that one. Just saying. It grows staph because, I mean, that's all you need to know. All right, biochemical tests. Essentially, like the stuff we were just talking about in that previous slide with, let's see if I can go back. Yes, I can. So when we're looking at this and it's talking about the color changes and the pH indicators, yeah. So we've probably learned by now that that's what we're looking at for the biochemical indicators as well. Some of it, right? So which test is this one? All right, so this test is the citrate test. That is the Simmons citrate auger that we put into a slant in our tests. So it has probably again going to be a question in a moment, but bromothymol blue is the pH indicator for this one. And we have alkaline shift if we are able to use citrate. It has to deal with the bicarb and ammonia situation that's going on in there. But yeah, it increases the pH. That causes it to turn that blue color. So blue is positive for citrate usage. Whoa, what? Okay. All right. Okay. Which of the tubes is positive? So for that test, I just said, oh yeah, look, be sure you look at the letters. Sorry. I should have had it not mix up. Yeah, so that's going to be B. So that's going to be that blue one. So I would be sure that you guys do remember like the actual color for the negative and the positive on those, because one of the things that people have an issue with the citrate and the malonate is that because they are typically green and they turn blue, most of the time you're going to turn the entire tube whatever color. So blue might take up the entire tube. So then if you're looking at a whole tube of blue and a whole tube of green, you need to know that the one that had growth on it and is positive is the blue. You can't just tell by taking up the whole tube, right? Some of them you can, but not always. Definitely not with malonate, right? Why is that tube positive? Why would we call that positive? Basically, what's going on in the tube? Okay, so that pH is going up in that one, so that's alkaline. All right, so that is Simmons citrate agar. How do we inoculate that one? Whenever we inoculated it, that was a slant, so we used our loop to basically streak the slant, right? How do we read it? You just look at the color change. There's no adding of drops or anything. Just looking at color change. What colors are produced? You're either going to have green or you're going to have blue. That's your choices, basically, with bromothymol blue. Green is negative or uninoculated, and the blue is positive. So that's what the colors mean. And the pH indicator that is allowing us to have this color change is going to be bromothymol blue. Let's see, next. All right, which test is this one? Why is this still here? Go away. So that's a tube, a whole tube. Okay, so this is a phenol red fermentation tube. And again, that's a partial picture of it, but I will say that for the lab exam, I'm not going to make you tell the difference between like a glucose or like a sucrose, like together. Which one is glucose? I'm not going to make you do that because it's ridiculous. So most of the time when you get a phenol red fermentation test, it might be as simple as what kind of test is this phenol red fermentation, carbohydrate fermentation. Or it might say, this is a PR glucose fermentation tube. What's going on in this? That's a more likely scenario. I think I have a couple of them that are set up that way. Just FYI. Okay. Notice how I'm not talking about the little tube inside. I'm pretty sure that's the next question. What is the name of the upside down tube that is inside of the assay tube? The test tube. Excellent. That is correct. Most of you guys got that one. That is the Durham tube. All right, this could be more than one answer. What are the two products of fermentation that we can detect from the PR fermentation assay? Excellent. So that is in fact going to be acid, which would be the yellow color change. So phenol red, it gives away what the pH indicator is here. That phenol red will turn yellow from the acid. And then the gas, we will see like actual air collected in the Durham tube itself, which will just look like an empty Durham tube. It should, if it is uninoculated, be filled completely with media. No, I'm happy with that. Remember, pink in the PR fermentation tube, that does indicate that there is growth and there's something going on, but it's usually just breaking down the peptone. Almost all of these media have peptone in there as a nitrogen source because we all need nitrogen to make DNA or amino acids or whatevs. So, yeah, so if they are just breaking that down, then that'll make ammonia as a byproduct sometimes. And then that ammonia is basic or alkaline, so it will turn pink. So it's not really indicative of what we're studying there. So which test is this one? Yeah, so this is going to be the SIM. So it's kind of divided amongst a lot of the different ones. So that's the SIM test. And of course, you guys will have the luxury of actually being able to handle the tubes and interact with them. So you're allowed to pick them up during the exam. So that helps you figure it out, I feel like, the hands-on thing. But what does the I stand for in SIM? Yep. So in the SIM assay, S is sulfur, I is indole, and M is motility. Remember, we're testing for sulfur with like a ferrous sulfate compound that's actually in the media. When the bacteria produce H2S or dihydrogen sulfide, if by reducing sulfur, then that will interact with the ferrous sulfate, the iron in there, and create a black precipitate. So black precipitate means sulfur reduction. Indole, however, is a byproduct of breaking down tryptophan. So the media has tryptophan in it. If your organism breaks down the tryptophan and has indole as a product, then we'll add something to it. And I'm going to get to what in a moment. And then let it sit for a minute. And if it changes colors, then that is indicative of the presence of indole. So it will be breaking down tryptophan. And then motility, that's going to be the stab in and out. In the previous question, it had that picture of like the 3 tubes. The first one was just like a regular tube. It looked kind of fuzzy. That's because they did the stab and it didn't have a clean line because it moved all throughout the media. Usually that'll make it look real like cloudy, or you'll see a lot of growth around the stab line. If it is nice, clean stab, and it's like even if they did it a wonky stab, but it still is clean and clearly visible, then that is negative for motility. How is indole detected in the SIM media? So what are we adding to detect the indole? This is the part that I left out on purpose when I was talking a second ago. Yep, so that's going to be Kovacs reagent. And you don't need to know the chemical reaction of how Kovacs works. Just know that we use Kovacs to detect indole. How do you detect H2S in SIM? Wow, I already just talked about that. I didn't realize. H2S is a product of sulfur reduction. Okay, so that is going to be observing a black precipitate in that media. So anytime there's like an iron additive that reacts with the product of the sulfur reduction, it can create that black precipitate. And that's how we know. Okay, how is motility detected in the SIM test? Wow, I went over all of them. Perfect, great job, guys. So exactly, it's gonna be growth away from that stab line. Look fuzzy or like just blurry. Okay, so with the SIM media, how do you inoculate that? We know it's a stab, because that's another one where we are looking for motility. Typically, that's going to be a stab. We're also doing the stab because it creates an anaerobic environment, which is necessary in order for us to get that sulfur reduction. Interpretation of the results changes, so the different color changes what's positive and what's negative for each test in here. It's 3 tests, so the sulfur reduction, the indole production, and the motility, what's positive and what's negative for each. And the addition of any reagents. In this case, we're talking about Kovacs reagent to test for indole presence, which is a byproduct of tryptophan breakdown. So which test is this now? So good job, guys. That is the litmus milk, that lavender colored milky liquid. In litmus milk, what color is associated with an increase in pH? Okay, so increase in pH. In this case, our pH indicator is litmus itself. So whenever litmus is exposed to an increase or alkaline pH, blue is the color that we'll see, from lavender to blue. However, if it is decreased, if it's acid, which I'm sure is a question that's coming up, then that would be pink. So that's the other side of it. So pink, lavender, and then blue, literally from bottom to top. Not in the tube, but on the scale. So let's see. Blue for that one. Alkaline is blue. In litmus milk, white. What does it mean? Yes, that's going to be reduction. Reduction of what? Of the litmus itself. So whenever the litmus itself is reduced, so it has this chemical reaction going on during the usually fermentation process going on in the milk. We reduce the litmus, it's no longer lavender, it's essentially clear or colorless, and now we see the color of the milk, so white. Okay, so we want to know the interpretation of the colors. The colors of litmus, you'll have the original lavender, you'll have the pink, which is acid, you'll have the blue, which is alkaline, which remember, alkaline means higher pH. And then the white, which means reduction. Okay, what is happening with digestion? If I were to be bringing that up, digestion is breaking down the casein protein within the milk, and it creates that sort of serum or translucent look to it. How is gas detected in this test? It will be breaking up of a clot or like air bubbles stuck within it. And what is the sugar that is in milk? I know we've said it before, and I know it seems like a stupid question, but just bear with me when I ask you that. So what is the sugar that's in milk? Lactose, right? All right, which test is this one? Okay, so this is one of the hydrolysis tests. We had 3 hydrolysis tests. It came with urease and gelatinase, and the starch or amylase test. So that was our starch hydrolysis. I don't remember if the next question has to do with it. So which media that has a lot of starch in it? Which organism pictured is positive for starch hydrolysis? All right, exactly. Great job, guys. That is the left organism. You saw on that picture clearly there was that halo around the growth because the bacteria had broken down the starch around it. So the iodine didn't interact with the starch there because there was no more starch. It was gone. So we see that halo develop there. We only had like two out of all four of my classes, only like two plates that were good examples of that, unfortunately. So what is added to detect starch hydrolysis? Let's see if you're paying attention to what I said. Yeah, so that's going to be iodine. It just reacts with starch and turns it like a dark brown. It's like a purpley brown. I say purple because on potatoes, it literally turns purple. All right, so which test is this? Test tube, clearishy, broth colored. It's got a Durham tube inside. All right, so this is nitrate reduction. That's what I have to say about it. What do reagents A and B detect? What are they detecting in the media? So you inoculated your sample, you put drops A and B. What are they trying to detect? So A and B are detecting nitrite. So if nitrite is there, A and B will turn red, turn the media red. And that means you had nitrate reduction into nitrite. So what does zinc detect then? You put your drops in, it didn't change a color, so you add zinc. Now, what are you looking for in the media now? Right, so some of you guys got this one. This is nitrate. So the zinc basically will turn it red if nitrate is still present. That means that the zinc had to turn it red. The bacteria weren't able to turn it red. So it detects the presence of nitrate. We started out with nitrate broth. Nitrate is still there. If after zinc you get red color, that's not nitrate reduction. You still got nitrate. So that's a negative test. So zinc is looking for nitrate. Remember, nitrate we test late. Zinc is the second one we add. That's the later one. All right, so know what is detected at each step with that nitrate reduction. I know that that one is a difficult one. I could go over it and talk through it a million times and probably just confuse you guys more. It is, I feel like, probably easier for you guys to look at like pictures and go through the pictures and what color comes from each one. So I'm going to leave that alone. But red is not always positive in this test. Be aware of that. Remember, the first step, we're testing for nitrite. And if it doesn't have nitrite, no color change, we add zinc because we're now testing to see if nitrate is still there. If it turns red then the nitrate is still there. That is no nitrate reduction. If it doesn't turn a color, that means we had nitrate reduction to, I don't know what. It could be a couple of things. You also could have gas production in this. The gas is typically going to be N2, although it could be other forms of gas. So they're going to be nitrogen related. If you had gas in your Durham tube, that is also a sign of being positive for nitrate reduction. All right, next question. Which test is this one? Yes, this is all one test. Exactly. So most of you guys got this one. This is the TSIA. It's the orange slant. All right, this could be more than one. Which tube or tubes shows evidence of H2S production in the TSIA slant? So H2S, sulfur reduction. Okay, so that's great. So we have sulfur reduction present in tube 3 and tube 5. That is really hard for me to tell what's happening with all these numbers on here. But you guys got it. Most of you guys got that one. These freaking numbers literally throws me off. Okay, next. Good job, guys. Yeah, it's that black precipitate. Again, same as with the sim, right? It's iron based. Which tube shows evidence of gas production other than H2S, which by the way is a gas? Okay, so a lot of you guys got this one. Yeah, a lot of you guys got this one. That's the 4A and the 4, the two that were lifted up. You could clearly see that there was gas underneath. The H2S tubes had little pockets at the very, very bottom. That's probably indicative of the H2S itself. So we can't necessarily attribute that to fermentation gas. It's the same thing as what we would see with the Durham tubes. But now it's just within the auger and it lifts it up like that. All right, so TSIA. TSIA, triple sugar iron assay, okay? Triple sugar. What sugars is it testing for the fermentation of? Which that's what it's testing for, fermentation of sugars. Glucose, sucrose, and lactose. So those 3 are the sugars we're looking at. So yeah, and then iron is the ferrous sulfate, same as with the SIM assay, that when it interacts with the H2S, which is the product of the sulfur reduction, it will make a black precipitate. So how do we inoculate this one? That orange slant is the one that we streak the slant and stab down into the butt of it, all in that one tube. How do we read it? We just look at the colors here, as well as the presence of gas. So we're not adding any reagents to this one. What colors are produced? Yellow. It could have no change in the color. That's indicative of things as well. It could have pink from alkaline production, usually from breaking down the peptone. So those are the, or black from the iron reduction, not iron, iron sulfate interacting with the hydrogen sulfide for sulfur reduction. So those are colors and the pH indicator in this. So we do have a pH indicator. That's why it turns yellow or orange or stays orange or pink if it is alkaline. That's phenol red. Okay, be aware of that. Phenol red, which tests out of all of the tests that we have done in this entire semester, which ones depend on phenol red as pH indicators? Which ones test for lactose usage? All right, which test is this one? Yeah, so this is going to be urease or urea hydrolysis. Which test is this one? So that's the catalase test. Everybody always thinks it is oxidase because it has to do with like oxygen. Don't get it confused, okay? If catalase, if we're thinking of like catalytic, I'm trying to say a catalytic reaction. So we're where it is I'm trying to look for here. Basically the enzyme that is helping out with actually breaking down. So remember, breaking down in this case, hydrogen peroxide, H2O2. We're breaking it down into water and oxygen, right? So if we're breaking something down in this reaction, which you have to in order to get the bubbles, right? Then that is like catabolic reaction, cat breaking things down, catalase, okay? The oxidase, that's cytochrome C oxidase. That is something that is in the electron transport chain of aerobic organisms. That's just a thing that is there. Okay, there's no reaction that it would be like necessarily that would be occurring. And we didn't learn about what the thing is that we put on the swab that made it turn purple. We just know we put drops on it, and if it turned purple, it had oxidase, right? That we say it had oxidase, it was positive for the oxidase reaction. It had cytochrome C oxidase. That's what that means. Okay. So that's what I'd say for that one. I don't know how else to help you guys keep from getting too confused between oxidase and catalase. What I want to say about urea, the urea test that was prior to this one, the pink and the sort of peach color, remember that there is a pH indicator there, and that's what causes that color shift. What is the pH indicator in the urea hydrolysis assay? In that broth, the pH indicator is phenol red. Again, there's only two, well, there's three, but I'm not really thinking about litmus. I guess there's three pH indicators that we ever learned about as far as the media goes, in like the tubes section, the biochemical section. Phenol red is the big daddy, right? Bromothymol blue is the green and blue one. And then we have litmus. That speaks for itself, I feel like. All right, good job, guys. All right, which test is this one? There you go. If we can remember. Yeah, so this one is going to be the oxidase. This one is. So we're just looking to see if this organism has cytochrome C oxidase. If we add in an actual reagent like hydrogen peroxide, and it's getting changed, broken down into water and oxygen, I'm just saying it again so that we don't forget, then that is a catabolic reaction. That is catalase. Okay, cool. I think we got that. All right, other tests that we did not go over in these pictures, malonate. Malonate is a tube, a tube. Go figure. It's a broth. It's a broth that is green, and if it is positive, it will turn blue, just like we saw with citrate, but it's like a liquid form of that. The pH indicator, which it is, bromothymol blue, okay? It turns blue when it is exposed to alkaline conditions. Again, malonate is broken down into alkaline products, essentially, that raises the pH and causes that green to blue change. All right, phenylalanine deaminase. Phenylalanine deaminase was like a typical looking slant, looked like any other auger slant that we could have had. We put growth on it. Then we put drops onto it. The drops that we put on it were ferric chloride. Okay, they're like that yellow, like almost oily looking drops. You put a few drops on there. If we saw a green color change on the slant relatively quickly, then that is positive for deamination of phenylalanine. Phenylalanine is an amino acid, deamination, removing an NH2 group, amine group. Okay, I'm not going to test you on that part of that. All right, the amine part. I'm saying it because chemistry. MRVP, methyl red Voges proscour. This is mixed acid fermentation. For the methyl red, you would add drops of methyl red And if you see that your sample is red, that is positive. If it is orange, that is not positive, that's negative. Your Vogue's Pro Scour was the one that sat there for 30 minutes after you added the appropriate drops, the alpha naphthol and the potassium hydroxide, whatever. It sits for 30 minutes after you mix it. And if you see red in the layer, then that is positive. Otherwise, it was like a golden color, right? We are testing for mixed acid fermentation with these. So fermenting carbohydrates and creating several kinds of acid as a product, not just like lactic only. Then we have gelatin. This was a hydrolysis assay. It was the first butt that we stabbed. It was the first butt that we ever stabbed, guys. Oh, so that one was a semi-solid media that if your organism broke down the gelatin or had the enzyme gelatinase, then even after I put it into the fridge, it would stay liquid because there's no more gelatin in there. It broke it down. So that would be positive. So liquid means positive for gelatinase or gelatin hydrolysis. Okay. So those are other tubes that we didn't cover right here. So this is everybody's favorite thing ever, the cells. I know people have such hard time with naming the cell shapes and stuff, so we'll go over this. How would you classify this organism's characteristic arrangement overall, in general? So this is a strepto arrangement. Strepto, strips, chains. Unless you guys got that. What is the name and shape of these bacteria? It's not the name of the bacteria itself, but the name of the shape. So here we're looking for the shape of the cell itself. This is a bacillus. Remember, bacillus is rod. Most of you guys got this one. All right, you want to know your shapes of your cells. Remember, shapes of the cells is a different thing than arrangement, so don't get those confused. So shapes of the cells, cocci or bacillus is all you're going to see on the test. Arrangements of the cells, you're really only going to see either strepto or staphylo. Strepto is the chains, staphylo is the bunching, or it might not have any arrangement, but I'm not going to ask you about that arrangement. And but that does exist. I just wanted to say. So just staphylo and strepto. Don't get these two confused. I feel like that is fair to apply for both of those situations. Don't get shapes versus arrangements. Don't get cocci and bacilli confused. Don't get staphylo and strepto confused. Those will get you through if you can remember that. Also, don't forget that simple stains are a thing. That a lot of you guys did actually utilize that at the end of your unknown projects to determine the shapes of your unknown cells, which was great. So you would probably remember what a simple stain is. But remember, we're just going to be heat fixing and then heat fix, and then you're going to be letting that cool, and then you're going to be putting your like stain on it. So I usually use methylene blue, but there are others you can use. It sits for a minute, then you rinse it off and look at it. That's it. You don't need all these steps. So remember that. Microscopes. You need to know usage and parts of it. So let's go over this. If you observe a specimen at 100x objective lens, and then a 10x ocular lens, what is the total magnification? You guys know this is going to be on the test, these questions. I'm sure you're aware of that. No, not too difficult, so I'm hoping that it's not a surprise for you. Yeah, so that's going to be 1,000. Most of you guys got it. Let's just multiply those together. All right, which lens is used with oral oil immersion? All right, yeah, so that's a 100x. Most of us are aware of that. No, again, so you're going to want to know the parts of the microscope and what they're called and what their function is on the microscope, not just what they're called, but also what they do. Lab safety, general, all of the stains, the tests, which includes the plates, that includes the tubes, maybe also includes Kirby Bauer, streak plates, ELISA. Those things are all fair game. And all the different kinds of media. And you will be given a little chart, like you guys had to fill out for your unknowns of like positive and negative for this test. I'm going to give you a whole chart that's filled out, and you're going to need to use a dichotomous key to figure out what organism is portrayed in that chart. So there's that. All right, yeah. So yeah, I'm sure you guys don't need any luck. Keep in mind, like I said, which tests have the phenol red in them, and which ones are testing for pH, or which ones are looking for lactose usage. If they have something in similar with one another, I would group them and try to remember which ones have those things and similar to one another, so that you guys, if I ask you which of the following tests have lactose fermentation, then you'll be able to check off whichever ones it is out of like a whole long list. So that's the idea with that. Okay. So a question like that is definitely going to be on the test, so be aware of it. Lactose. Okay, I think that's that. Okay, do you guys have any questions for me other than this? So I'm going to consider this like that's it. We had the review for the lab. Hopefully I've covered all the topics that are going to be covered and that you guys need me to cover. If there's anything you would like me to touch on more at this point, speak now or forever hold your peace. Well, not really. You can always email me. So if you guys have any questions, let me know. Otherwise, I'm going to call this quits. Oh, I should put your podiums up. Here you go. Sorry, I'm a bad teacher. Here we go. That's better. All right. If you guys don't have any questions, like I said, email me if you guys get deeper into studying and you still don't understand something. You can email me too, and like I'll do my best to get back to you, of course. And if you needed to talk to me on Zoom or something to go over or something, email me and cross your fingers that I almost always check it. Hopefully I can get to you before the test. It is set up and ready for us to take it tomorrow. So we'll do lecture, and then whenever we get done, we'll just go up to the lab and get started with the exam. If you come in late, that's okay. For those of you who are my Monday morning people anyways, that's okay. Remember, you just still have until the end of lab to get done. It'll be more than enough time. You guys know that now. So yeah, cool. All right, guys, I'm going to cut you off here. Okay.